Welcome everybody to 52 Living Ideas. I'm delighted to have Sanjay back to do his neuroscience series. And today we're talking about what thoughts look like in the brain. So Sanjay, take it away.
Thank you. So first, I just wanted to go and I see a lot of new people here, and I wanted to just give a very brief background. I mean, I'm not a neuroscientist, but I work with a lot of teams. I work in the biological sciences. I've worked in for my entire career, and a lot of the work that I do has to do with many areas of biology, including neurology. And some of the teams that I've worked with, they have been doing research in neurosciences. Other teams that I've worked with have done research in other areas. But this is an area that I think is interesting for most people, and as well as for me. And so I wanted to go into this in more depth. And I find it very interesting because the questions that we get asked in these sessions are very stimulating. They actually make me think a lot more than the things that I learned. So that's an aspect that I absolutely enjoy here. So today we're going to talk about something that's pretty basic in when we think about our mind, our brain, and that is this concept of thought. And just to clarify, from the perspective of neuroscience, or at least from the perspective of how the brain creates a thought or holds a thought and an emotion are pretty much the same thing. There's really no difference between them. So everything that we talk about today is really geared around thoughts and emotions. I won't use the word thought and emotion, but we're talking about both of them. They're basically interchangeable. The only major difference is that emotions tend to occur primarily in certain regions of the brain, whereas thoughts are in other regions. But oftentimes, what we won't go into this today, but what we find is that a thought and emotion, they coexist. And oftentimes they move from place to place. So when a thought originates, or let's say an emotion originates in some of the emotional centers, for example, the mesolimbic area of the brain, it will move to other areas. It may move to the temporal lobe, definitely, and then it may migrate into the frontal lobe. And at that point, it becomes more of what we might think of as a thought, but it will still have parts of that emotion connected to it. So there's really no reason to differentiate the two. They're really the same thing. It's just, we use it linguistically in different ways, but in terms of the neurobiology of it, there doesn't seem to be much difference between them. So I have some slides that let me go into. So hopefully is this visible? I think this should be visible, right?
Yep.
Okay. All right. So first, what we'll do is we'll start off with just the very basics of things around a thought, right? So what the anatomy of a thought, where it occurs, how it occurs, things like that. Also, what structures are needed in the brain? What is it in our brain that allows us to have thoughts? Now, one of the things for those who are new here, we've actually gone into many, many different areas. So I'm not going to go into a lot of these, repeat a lot of the ideas that we went into, but I may touch on them and may repeat some of the key points. But we're not going to go into detail on a lot of these other areas. And then basically the brain is a network of neurons. This is, I'm sure most people who are attending understand this, but it's actually this concept is fairly extensive, and it pervades through every area of neuroscience and actually affects pretty much everything that happens and how we look at all activity within the brain. So the brain is basically a network of interconnecting neurons, right? And the mind is the interconnections, connections between the neurons, between the nodes, and the electrochemistry of the signaling between these neurons. So these together, in a very basic way, these together are what create activity in the brain. And one type of activity is a thought or an emotion. And then there's something called graph theory, which is actually an emerging area of the neurosciences. It actually has interdisciplinary interconnection with many other areas, mathematics, definitely computer science, other areas of biology, but this is something we won't go too much into this. I have 1 or 2 slides on this that we might touch at the end, but this is an important area. And if this is something that I think we need to, then I may go into it much further in the future, but I'm just going to introduce it for now, later in the talk. So the first thing we'll do is we'll look at basically the anatomy of a thought. What is it in the brain? And for example, what areas, where, what is a thought? What areas do they occur? How do they occur? So starting off, what is a thought? So a thought basically, and let me preface this by saying that a lot of the talk tonight is not about what is definitively known in neuroscience. These are areas that there are actually a lot of understanding, but the details of I'm going to go at the top level, at almost a superficial level, though a lot of what I'm saying has been confirmed through a lot of research. And hopefully most of the attendees, if not everybody, saw at least one of the videos. I posted 2 videos. One was about 20 minutes long, and hopefully everybody saw that, because that video is very important to really understand what we're going to talk about. And for copyright reasons, I don't post the videos themselves. I might have snippets from parts of it, but it's really, I would hope that people have seen the video, because it will help you to understand what we're talking about. So a thought or an emotion is a large group of neurons that are activated together. And these neurons can be in different areas of the brain. They don't have to all be in the same area of the brain. And we'll go into this a little bit later. And if you saw the first video, then you already may understand a little bit of this, but we'll go into a little more. And when they're activated together, the word together I have in quotes because they don't have to necessarily be activated simultaneously at the same instant. They can be activated within a certain time span of a few seconds within each other. And together they constitute a thought. So a thought is kind of like a moving state. It's not a static state of the brain. So this other concept is that of a brain state, and that's something that's important. And we'll go into that to explain some of that, because that is very important to understand exactly what a thought is. Next, so let me just do a very quick, so we have a little picture of an icon of a brain, and this is a little pictogram of 36 neurons. Okay, and neurons are not this homogeneic, and they're not disorganized and in a grid. They're very, very random and dispersed. If we had 36 neurons, they would be all scattered, and you would not. Some of them, and also neurons exist in three-dimensional structure. So some of the neurons, if you're looking at them through, let's say, if you're looking at neural tissue on a slide, your microscope would only focus at a certain depth. So you won't be able to see all of them. You would see a certain layer at one depth. So these 36 neurons would not all be in the same focal plane. But to simplify things here, we've done, use these pictograms to simplify it. So this pictogram has these little lines, you might see very thin gray lines connecting them. And some of the neurons are turned on, you might say. Some are bright red, others are medium red, and others are pinkish, very light. And this is an area which we're not exactly sure about. We know that neurons activate at different levels, but we don't know the importance of that in terms of brain processing. Whether there is simply a threshold above which the neuron is considered activated, and everything below it it's considered inactivated, or perhaps neurons have multiple levels of activation. For example, neuron might actually activate at 3 or 4 different levels, and depending on the level of activation, the signal from that neuron gets sent to other neurons in a stronger way or in a more attenuated way. So these are things that we don't know yet. So I'm giving a very simplified model showing only a binary state of neurons that they're either on or off. But in this diagram, I've actually shown tertiary 3 states. So this cluster of 36 neurons, let's say that it's it's actually resides in the occipital lobe, which is the back area of the brain, and that's where our visual processing occurs. And let's say that this cluster of neurons, the configuration of these neurons being on and off, connotates the concept of blueness. So the person that has this brain, their eye is seeing, let's say, blueness. Okay, they might be wearing sunglasses that colors the entire field blue, or they might be looking at a room that is predominantly blue, or in some sense, they have a sense of blueness. So this is an example of what the concept of blueness. Now, blueness is not a thought. It's only a fragment of a thought, right? But we're going to build up on this. So the concept of blueness is encoded in a specific area of the brain. And this is something that we recognize today. And if you saw the first video, you may recognize that. That video talked about concepts such as physical objects in the world that the researchers described. And so there are physical concepts in the world. There are concepts around our own body. There are verbs and processes that we encode. And there are interrelationships between us and the world. These are types of things that we encode in specific regions of the brain. And I'll go into some of that. But I think in the question and answer, we may also go into some of this. So this is an example where, how neurons are encoded in the brain, and a specific region of the brain may encode a specific idea. Okay. Now, one thing that you might notice is that in this set of neurons, there are 36 neurons, but some of the neurons are not activated at all. And that's actually important because out of these 36 neurons, roughly 12 or 13 of them are activated, and the remainder are not. And this pattern of all of the ones that are activated strongly, including the ones that are activated to a medium level, and the ones that are activated very mildly, as well as the ones that are not activated at all of these neurons together form the state of this cluster. So all of these together are required for the concept of blueness. If the bottom half of these neurons, of the neurons that are white, if they were somehow activated, this may end up being a completely different thought. It may not be blue. It may be partly related to blue because in the upper right, we have this strong cluster of neurons, which perhaps have a component which may characterize a subtle element of blueness, but it's not the complete idea of blueness. So if there is another cluster of, within this cluster of neurons, if there are additional activation of other neurons, it may actually mean something else to that specific person. So, again, this is a simple idea. The next idea around thoughts and emotions are where can they occur? So thoughts and emotions are part of our higher order thinking. They occur within the cortex or neocortex, and also the mesolimbic areas, which are where emotions predominantly originate and reside. They are not in the hindbrain, which is the cerebellum, the pons, medial oblongata. We've talked about those various times. Those areas of the brain are not really thinking. They're more primitive areas of the brain, and they're responsible for much more primitive aspects, such as basically keeping our body alive, homeostasis, various other aspects of the cerebellum is involved in motion and proprioception and other areas of our physicality. These areas are not involved with actual cognitive function. So the cortex, the neocortex, is the main area that has thinking, and then the limbic system is involved also. So how do thoughts and emotions arise? Okay, so that's a question somebody may ask. And they appear due to either external stimuli or internal stimuli. So for example, our sensory inputs, our ears, our taste buds, our eyes, obviously, they sense a lot of stimuli from the world around us. And when we're seeing something, almost always when our eyes are open, we're seeing things. So that forms stimuli that goes directly to our occipital lobe. Again, the example that I gave earlier, we may sense various colors in the real world because the world is multicolored. There may be multiple color regions of our brain, multiple color detectors in our brain that may activate simultaneously, as well as, so just looking at the visual center, the most primitive level of visual processing may involve the detection of colors, especially the strongest colors that are in front of us, as well as patterns and objects, things like that. So the visual cortex will have many, many types of these small regions, similar to this blueness region that we looked at earlier. But there may be a region for, let's say, if you're looking at a dog, there may be a region for fuzziness or furriness, because dogs have furry hair. And there may be, there seems to be a region in our brain which revolves around furry objects. There may be another region that has to do with smoothness. So if there's a dog and there's a smooth object next to the dog, that furry region and the smooth region both may activate together. So this is an example of how the stimuli that we get from our senses cause activation of clusters of neurons within various parts of our brain. In this case, we're only talking about the occipital lobe, but this can happen, for example, when we're active, we're also hearing things. We're also listening to audio or sounds, and that will activate similar clusters within our temporal lobes, both on the left and right side. So, all of the stimuli that we experience in real time are causing activation of various clusters. These clusters, and the clusters may be larger sizes. Some clusters may be very, very small. So that's one case where we have stimuli from the outside world coming in. We also have stimuli from the inside, from our body itself. And there are actually two sorts of those. One are emotions and thoughts that already exist in our brain. And the other may be sensations within our body. So our somatic sensory system will take sensations from our viscera, from our abdomen, and for example, if you're feeling nauseous, or if you're feeling scared, or if you're feeling frightened, right? These activate. There actually are neurons in other parts of our body, much less than in our brain, but those neurons will activate, and they'll activate our parasympathetic and sympathetic nervous system, and they may actually communicate to our brain, at which point our brain becomes aware of these sensations in our body. So once we have these sensations coming from outside and inside, they will coalesce in various regions of our brain, and together they will evolve into what we call a thought or an emotion. How long do thoughts typically last for? Typically between 10 and 30 seconds. That's for most of the case. They can be longer, but to be longer, you actually have to actively try to keep the thought in your mind. You have to take some active action to either remind yourself or to do something to keep that thought in place. So if it's a thought that is let's say it's an emotion, emotions also will subside and almost disappear, or at least from our consciousness. Excuse me, they'll alleviate after a certain time. But if you keep energizing, if it's emotion that is, let's say, either a happy emotion or a sad emotion, you can recreate or re-energize that same emotion and keep it in your mind for longer. But that's requires additional steps. That's not normal. Also, thoughts and emotions can propagate. They will move from region to region, not as a distinct thought, but if, for example, the concept of blueness. The blueness is a fairly simple thing. Let's say the concept of cold, right? The temperature cold would originate in the somatosensory area of our brain, which senses temperature, right, from our skin. And that area of our brain may sense coldness, right, temperature. And that thought that sensation of coldness may actually move and expand into other areas of our brain, especially in our frontal lobe. And if it expands into our frontal lobe, it will become conscious. So what this means is we can actually have a sensation of a thought or emotion or an idea that we're not conscious of, because they're in other areas of the brain and they're not focused on by the prefrontal cortex. Once they reach the frontal cortex or the prefrontal cortex, depending on the type of concept it is, we probably will become aware of it, and then we may start to feel the sensation of coldness. And at that point, we will not only feel the sensation of coldness on our skin, that may actually pervade the scene around us, and we may feel that the environment around us is cold. So a lot of times when you're in wintertime, when you're walking outside and you have your jacket, let's say, unbuttoned or open partly, you may feel a little bit of cold air on your chest. And if there's a gust of wind, it may send a chill through you, right? But that chill takes time to propagate through your brain. In the beginning, that chill will hit the sensory areas, and the skin surface of your skin will feel it first. And only the somatosensory areas of the brain will sense it. But over time, over a few seconds, usually 2 or 3 seconds, because that's something pretty rapid, it may move into our consciousness, at which point we realize what happened, because we're not seeing the air move. We're not seeing coldness move into us. We don't see something touch us. All we sense is a sensation of coldness in our chest. Then what happens is our frontal cortex will consider the possibility of what this means. And it'll start to think about it. And it'll start to realize that my jacket is open. I'm outside in the cold. Wind can occur. Probably wind went and it sent a gust of cold air along my chest. And now I'm sensing that coldness. And then after that, you may actually have a sensation where you start to feel your entire body starts to feel cold. Even though only a certain part of your chest actually felt cold, the sensation will move into other areas of your mind, and then your whole body will feel cold. So this is an example of propagation of thought. Another type of propagation is when a thought can actually turn into another thought. So you might have a thought, you might have a stimuli of, let's say, a slice of pizza. You're looking at a slice of pizza. And after that slice of pizza, and that's not a thought, that's not a complete thought, you may start to think about what you had for lunch. And then that thought of lunch may actually turn into another thought for what you're going to have for dinner, right? Because that's a completely different thought. The thought of what you had for lunch is a thought of a past activity and objects in your past, objects that you ate. And that can turn into a different thought, which is a thought about the future and about other objects that you might eat, right? Or objects that you don't know what you're going to eat because you don't know what you're going to have for dinner. So this is an example where thoughts can turn into other thoughts. And this happens all the time in our mind. And this is also another reason why, the 10 to 30 seconds that described here is only for an initial thought. If it changes it to another thought, obviously that other thought can stay for another 10 to 30 seconds. And the numbers here aren't written in stone. It's variable. It depends on the situation. It depends on the dynamic nature of the environment you're in. If you're in a very fast changing environment where many stimuli are coming at you, then you probably won't sense, then your thoughts won't stay for 10 seconds even. You might have thoughts that stay for 2-3 seconds and then they turn into other thoughts. So now we're going to go into a little more of the first step of that. So here, what we're going to do is we're going to look at the concept of what is a brain state, because that's central to what we're talking about here. So here's an example of a brain, which, and in the simplest sense, if you think of a brain as a single neuron, okay, it is not. It's billions of neurons. But if you simplify it to the most ridiculous state that you could of a single neuron, that single neuron can be either on or off, right? And that's known as two states. Okay, so this single neuron would have two states, and some of the states, if you think of that neuron as being off, you might think of it as the person is asleep, or they're in a coma, or maybe they're dead. Okay? So that's the equivalent of that neuron being off, or the brain being off. And the other state is that of consciousness, or awakeness, or being alive, and that's the state of on. So in this simple example where the brain is just a single neuron, there are two states. Okay? So there are two, you might say, configurations of that single neuron. Now, the brain has many more than one neuron, so that's where the concept of state becomes a little more elaborate. And so next slide, we're going to go into a little more of this. Let's take a different example. Here's a picture of a very simple room, and there are 3 light bulbs in this room. And this is an analogy for a brain. So this room is like our brain, okay? And there are 3 light bulbs are like 3 neurons. So let's convert it into what we're more familiar with the brain. And there are 3 neurons here in this brain, right? In this case, each neuron again can be on or off. So what this ends up giving us is if all 3 neurons are off, that's one configuration of this brain, right? And again, this brain is very simple. It only has 3 neurons. Another configuration is where all 3 neurons are on, right? So those are 2 configurations of the brain. And then if we fill in the details, it actually gives us 6 more configurations. So there's a total of 8 configurations in this brain, okay, with only 3 neurons. Okay? And what happens is, so this is an example, again, at the beginning I talked about the simplification that we made, that neurons can only be on or off. In reality, we believe neurons actually have they're multi-state, but we don't know whether it's important, whether the activation, the propagation of signals happens at multiple states. For example, if a neuron has 4 states, it's possible theoretically that after it reaches the first threshold, it may activate some of the closest neurons around it. After it reaches the second threshold of activation, like it becomes half on, it might activate a lot more regions in its neighborhood. And after it reaches a third, it might activate more. After it reaches the highest level of activation, perhaps full activation, it will send its signal to all of the neurons that it's connected to. Okay? This might be one version of how neurons function. Or it might be as simple as just on or off, that if it's off, it doesn't send any signal out. If it's on, it sends a signal to everything that it's interconnected to. So these are two different examples of a single neuron, how it might work. We really don't know right now. But we know some of it. What we know is that in some areas of the brain, neurons seem to have multiple levels of activation. Other areas of the brain, they seem to have less. And it's possible, theoretically, that within a certain region, there may be multiple of these types of, in other previous talks, we've talked about multiple types of neurons. And so depending on the type of neurons that exist in an area, this activation profile of the neurons will get much more complicated. So in this simple example of theory neurons, we have 8 permutations of just 3 neurons. So if we expand this brain to have, let's say, 11 neurons, okay, just picked 11 out of thin air, no particular reason for it. What that means is 2 to the power 11, and 2 here has to do with the 2 states the neuron can have, either on or off. If each neuron had 3 states, it would be 3 to the power 11. If each neuron had 4 states, it would be 4 to the power 11. So the number of states increases exponentially. It gets very, very large, very, very rapidly. So just with 11 neurons, if you would have a matrix of 11 neurons, you would have 2048 possible configurations that all those neurons can be in, either on or off. And again, configuration state is another word for a configuration. Now, in the previous slide, we had 36 neurons. If we look at an example of just 30 neurons, that gives us more than 1 billion states. So you have to, and 30 neurons is a tiny, tiny number. Our brain has around 86, adult brain has around 86 billion neurons. So 2 to the power 86 billion is an immense number of neurons. It's actually more than the number of atoms in the observable universe that we know of. So it is such a large number. It's I won't use the word astronomical, but it's a very large number. Another point that's important here is that out of the 2 to the 11 neurons, out of the 2048 states that these 11 neurons would have, typically every neuron is not necessarily activated all the time, right? Typically what happens is if you have, and for example, in the entire brain, out of the 86 billion neurons, all 86 billion neurons don't get activated or deactivated based on stimuli. Stimuli only affect certain regions of our brain. And typically the regions that they affect are in the order of several 100 million to a few billion neurons in size. The cluster size range from a few million, dozen million cell neurons to perhaps 1 billion or more cells. The cluster of that size is really what we're talking about when we're talking about thoughts and emotions and neural activity. So at that level, so if you've seen, most people I'm sure have seen fMRI images of the brain. And when you see these images, you see color coding, oftentimes they're color encoded images. And there are regions of the brain that are one color that are activated brightly and other regions that are activated less. Oftentimes these regions that are typically 2 to 3 centimeters in physical size compared to a normal sized brain, about 2 to 3 centimeters in diameter, is typically probably about 100 million to 1 billion neurons. And again, it depends on the density of neurons, because the density depends on the area of the brain we're talking about. So these regions we're talking about can grow, to immense size. Here's an example of, taking the example we had earlier of blueness. Okay, we're going to expand on that. And so let's say a person has a concept of blue. Now here, so what I want people to understand is we're taking this in slow motion. Okay, in the real world, in an actual brain, all of these things happen within several hundred milliseconds. So within a quarter of a second, a thought flashes into our mind So when we see a scene, and the scene may have some large blue object in front of us, okay, in the first few, perhaps in the first 50 milliseconds, our brain will get a sense of blueness. Okay, so I'm doing this in slow motion. This is a slow motion version of a complete single thought, although it is a simple thought that we're going to start with. So in the beginning, when you're seeing something blue, you'll have this concept of blueness, okay? But you won't even recognize that it's a color, okay? After a few additional 100 milliseconds, you'll have a realization that you're looking at a color, okay? So these are two distinct concepts in the brain, the concept of blueness and the concept of a discrete color, because the color talks about a discreteness, right? Blueness may be a spectrum. Blueness may have to do with a range of colors that go from purple to ultraviolet. And it may be a triple. It may be from the green area to the purple area to the ultraviolet area. This triad of colors would encompass blueness. Okay, it doesn't talk about a single color. But then when this concept of a color comes into mind, that solidifies it as if we're looking at kind of like a single color. So that's another, I wouldn't say thought, it's another precept that arises in the brain as we look at something. And then, you might have another concept that arises because what's happening is you're looking at it from your eye. Okay? And there's a somatosensory area of our brain, which is along the top. There's a sulcus, primary sulcus here, which on the side closer to the rear of the brain is related to all of the sensations that sensory inputs from it is from every area of our body. And then opposite to that sulcus, on the frontal part of the brain, it's at the rear of the frontal lobe, is the motor control areas of that. So in this example, the eye is active, the eye is doing the observing. And so there will be an activation of, you might say, clusters that have to do with realization that the eye is active and the eye is somehow moving. So you might have a realization that there's blueness around you, that there's a color of blueness around you, and that your eye is involved somehow. And this the fact that this entire area has to do with ourselves, that's why the concept that I wrote here is me and I together, because the concept of my eye, or this entire somatosensory area, is also an area where we encode our own body, aspects of our body. So we know through, from the time of birth, after birth, we've learned that our eye is a physical object of our body. So the concept of me and the concept of I is combined together, just as the concept of our hand and our eye, our hand and me is also combined. Any individual part of our body and the concept of me is combined together. And there actually seems to be other concepts of me, which is more toward the frontal part of the frontal lobe, which is a distinct, a more metaphorical concept of me. This is a physical concept of me, okay, that I described here. The metaphorical concept of me as a virtual me, or me in the future, or something like that, would involve a different region of the brain that would be probably more to the right of this that we're looking at here. And then we might have these come together into the prefrontal area, and these three regions might come together, and they might. So the frontal area is where our consciousness, our conscious awareness is. So this might come together to give us this sensation that I see blue color. Okay, this would be a thought that you might have. So this is the formation of a thought. If you're a young child, let's say, and you see a blue color. Now this may be after you've had language, because this is something we might touch on, because language is one of those things that's very fundamental to thoughts. If you don't have a sophisticated language, the sophistication of your thoughts are also limited. So proportional to the complexity of language that we have, and in one of the earlier talks we talked about, for example, just the concept of color, right? So the more colors that certain languages have, the more complexity we have around the descriptions and the vocabulary of color, and this also has to do with this ability to actually sense these colors. If a certain society has doesn't have certain colors in their language space, those people actually are not able to discern those colors. They actually don't see those colors in a sense. Perceptually, their brain observes it, but the concept of blueness here is a thought, right? That's encoded. They probably don't have an encoding of that color because in their language, in their experience of the world, they haven't given importance to it, so they have not encoded an area for that specific color. So that's an example where the richness of a language is actually also tied to the development of these sub-thought-based concepts that we encode. So this thought of I see a blue color is something that most everybody would experience, even if they're a child, because this doesn't require complex language. Now we're going to take this and look a little bit deeper. Okay? So let's take the same concept, blueness and color and eye, right? And let's add to this. Okay? Now what we're doing here is we're talking about complexity of thought. Okay? So the original thought was I see a blue color. Now we're going to add something. Okay? Sorry. You know what? This is not working. Okay, so I'm not sure. So the concept here. Sorry about that. I should have done it. So the concept here is that there was a blue banana that the person also saw. Okay, so let me just walk through this. This is just moving. I don't think I can pause it. Sorry about that. So there's a blue banana that the person saw. Okay, so there would be encoding of a blue banana, not a blue banana, but a banana. Okay, and then there's another thought. There's another precept that they had of their left foot. Okay, and the left foot would be somewhere in this area. So all of these together would come together and form this concept of I see a blue banana near my left foot. Now the similarity with this and the previous thought that we had is that this thought and the previous thought, in a sense, this thought the previous thought is superimposed on top of the thought of the precept of a banana and my left foot, right? So if you think of another little box over here of a banana and another box over here of my left foot, right? These four are the thoughts that we had from the previous slide, just of seeing blue, right? And so when the brain senses all of these together, and the frontal lobe, what the frontal lobe does is it actually interrogates, it kind of goes back and checks out what's going on in all of the other parts of the brain. When it sees activation of these other parts of the brain, it brings those activations together into a unified concept here. And probably what is necessary here is some type of rudimentary language, because once we get into more complicated thought, for example, if we simply have concept of a color and blue and eye and foot and banana, okay, those 5 things can be put together into multiple sentences, right? You can have I foot a banana near my blue. Okay, that can be a valid construct out of those 5 areas of the brain, right? Or you can have blue banana sees my near foot. Okay, that's another construct. But the fact that we see it as a sentence, a coherent sentence, is because we have language as a learned area. The language is basically in the Warnock's area, which is in the temporal lobe region, near the center part here on the surface, but it's near the central region of the brain. So the fact that these ideas, these precepts come together in this order, the ordering comes out of the language lesson that we have, or the ideation that our language skills allow us to have. So that's also very important. I didn't go into that here because that will make this much more complicated, but language seems to be pretty important with how we put together these constructs. And depending on how sophisticated language you have, you can come up with much more complicated sentences and ideas. And if you had, let's say you have 20 different areas of the brain that are activated together, you can form a very, very complicated sentence, which you'll be able to hold, although a sentence that complicated, probably you won't be able to hold in your mind for that long, because you have to keep activating each of these different areas over and over and over. And if you have 20 of these areas to keep activating, you're probably going to be able to activate only 3 or 4 or 5 of them. We say that typically we can activate only, we can remember only about 7, between 5 and 9 things in our mind at any given time. And this is an example where these individual concepts are things that we have in our mind that we need to keep activating. So if the concept has more than 5 to 7, 5 to 9 concepts, it becomes very difficult for us to keep all of those areas of that idea within our mind simultaneously at the same time. That type of very, very complex idea may actually evolve, and we may understand.
Looks like there is a problem.
Half of the time, they're bringing a different.
Sanjay, it looks like you're frozen. Give me just a second. Folks, are you also noticing? Okay. All right. Sanjay, are you back?
You tell me.
Yes, I think you're back. You're back.
I don't know if that was.
You just, I think you lost. Just repeat what you said for the last 30 seconds.
Okay. So the example I was giving was of a more complex thought, a very complex thought where we might have 20 different regions in our brain that are activated. And typically what will happen is our prefrontal cortex has to activate all of the different areas of our brain. And because we can only remember roughly 5 to 7, 5 to 9 things in our brain at any given time, what our brain ends up doing, or what our frontal lobe ends up doing, is it activates parts of that complex thought. So it might have only half of that thought in mind at a time, of a very, very complex thought we're talking about. It might understand only a half of that thought at one time. And then it shifts and understands a few other of those, out of those 20, a few other of those precepts. And it becomes aware of that other half of that thought. And then later it shifts to another set of those 20. So it might go between 6 of those precepts and then another 7 of those precepts and another 7 of those precepts. So 6 plus 7 plus 7 becomes 20. So it might take the brain, it might require the brain to cycle through these 6 and 7 and 7 over and over and over to keep all 20 of those thoughts, this very, very complex thought in mind. And this is one of the reasons why very complex thoughts are very hard for us to hold in our mind. And oftentimes what happens is we simplify very complex thoughts into just a few things. Most people do this because it is very, very difficult for us to have that simultaneously. This is just a fundamental limit of our brain. So sorry about the animation problem here. So going more into thoughts. So the next topic we'll go into. So those are just examples of how thoughts arise in our brain. So
Sanjay, you need to restart your sharing screen. It went off.
Let me, okay, I think that should be visible. Okay, so let's look at the next idea around thought is how, so what makes a thought, right? So how are thoughts formed? So thoughts require millions and billions of neurons. Right? And again, there are regional variation in the types of neurons. There are variation in the density and morphology, the structure of neurons, how they exist. And in the outer layers of the cortex, in the gray matter, in the dura matter, not dura matter, in the gray matter, neurons exist within 6 layers in the human brain. And so it's a 3-dimensional structure. It's roughly 4 to 5 millimeters thick. And so this 3-dimensional structure has very different morphology. So the structure and density of neuron, and within these 6 layers, the density varies, the types of neurons vary. If we're talking about a different area, let's say a hippocampus. In the hippocampus, there's very, very different types of neurons. And again, the density and the structure of the neurons, the way that they're positioned relative to each other, also is very different. So depending on the part of the brain that we're talking about, there are very significant changes and differences in how these neurons grow and exist. Also, other than density, we have interconnections. So interconnections, and again, we've talked about these things in prior talks. So the interconnections are basically the axons, where we talked about every neuron has an axon and every neuron has dendrites. So the three most simplest parts of a neuron are the cell body, which is the soma, the dendrites, which are what are used to obtain stimuli or impulses into that neuron, and the axon, which is you might say the output, which sends the output of that neuron to other neighboring neurons. So the interconnections that we're talking about here, the interconnections are these axons, and axons can be anywhere from a few micrometers, if the neurons are very, very close, to several centimeters long. There are axons that are within our brain. There are axons that are 5 to 6, even 8 or 9 centimeters long, which is significantly. For example, the axons that connect from the left hemisphere to the right hemisphere, they tend to be multiple centimeters long. The axons that connect from the front of the brain to the back also tend to be long. And the center of the brain is actually filled with these interconnections. So if you had in most MRI images, it is there, but axons don't appear very readily in fMRI images. But if you had, let's say, a CT scan, depending on the opacity, but axons will appear in some sense, but they have a lot of fat molecules in them, a lot of lipids. So they will appear, but they'll be more cloudy. But the central mass within our brain is all of these interconnections. They're basically very, very thin wires, but they actually occur in bundles. They're not individual nerve. Many of the nerves that go from one region, they after they reach a certain area, there's a additional layer of myelination sheathing which actually it's almost like electrical wire. If you think of electrical wire and a bundle of wires that have another sheath around them, something like that happens with our neurons, with our interconnections also. So the interconnections that are long distance, multiple centimeters long, actually have that type of protective layering within it. So you have axons within bundles of axons. Those bundles of axons are grouped together, and there's an entire sheath around multiple bundles of axons. So those are the major tracks and pathways within the brain that these signals propagate from one major region to another major region. So these interconnections are very important. And then within a region, you have many smaller, thinner, shorter axons which communicate within. And then you may have neighboring regions which don't necessarily have these thick axon bundles. They have other types of interconnections. So interconnections are very, very important. Also, we have Golgi cells and many other types of cells in the brain, which are not actively involved in thoughts, but they provide support for our brain. They also absorb neurotransmitters, and sometimes they may emit or reconfigure neurotransmitters. So there may be some activity that they're responsible for that affects how our brain functions, including thought. We're not really sure of what else may be involved, but there may be other things involved with thoughts. But a high density of neurons and a high density of interconnections are two things that are very, very important, as well as the variations among the density and the morphology of neurons. So the myelinated axons we talked about, but I want to go a little more into this. So what a myelinated axon is an axon which has, there are, depending on where in the body, there are Schwann shells and other types of cells that basically coat and cover these axons, which is you can think of it as a wire, which sends the signal out. And it's coated in myelination. And what that does is it allows that particular axon, which is myelinated, becomes much, much faster than an unmyelinated axon. So many of the long distance axons are myelinated. Most of the long distance axons are myelinated, which allows the signal to propagate very, very quickly. We're talking about a few milliseconds. So this allows faster interconnection between regions. Some are very long, multiple centimeters long. We talked about that. And they connect pretty much every region of the brain with every other region of the brain and between the hemispheres. So this is very, very important in our mind. Also, neurotransmitters, glial cells, and there may be other things that again we may not be aware of that are actively involved. We know there are many other things that have passive activity, but there may be other things that actively influence the generation of thoughts and emotions. Also, we talk about language. Language seems to be, this is something that probably needs a lot more research, but there have been some studies done, but language seems to be something that helps with more complex ideation. And the example we talked about in the previous slide is that the simpler idea of I see blue or I see a blue color, really you almost don't even need a language to understand that, right? If you can think of a child who doesn't have language yet, when they see an object that's a blue color, they'll have a sensation in their mind of blueness, and they'll have a sensation that they're observing it. Like they might move their hand, and that object moves along with their hand, this blue object. And they'll have the concept of, this is a blue object that's moving around, so I see this blue object. That they might have a concept of that, even though they don't have language at that point. But to have much more complicated ideas, I think language is probably a much more necessary example. So let's take a break here and do some questions and answers.
All right. So folks, it's time for Q&A. Sanjay, I'm going to mute you when I'm speaking because there is an echo. All right, folks. So it's time for Q&A. Go ahead. The rules are very simple. As always, go ahead and type exclamation mark in chat or raise your hand in Zoom. Questions only at this point. Try to keep them brief and on topic. And feel free to disagree with anybody on anything. Just do so courteously. So it's going to be Joe, Claudio, and Kevin next. Give me just a second, Joe. Hold on. All right. Go ahead, Joe.
Hi, Sanjay. Great presentation thus far. So thank you. This is somewhat of an easy question. Just a basic question is that you talked about, how the neurocluster of, what made blue, or what made us in the brain process blue. What is the experience, or why is it that people that are colorblind process something different? And what are the implications to that? Like, what is the distinction and why does it happen?
Yeah, that's a very interesting question. And that actually has a lot of additional repercussions to our understanding from that. So first, just quickly, when we talk about the concept of blueness and how that arises. So when a baby is born, the baby doesn't know anything about any color. It doesn't even know colors exist. It's simply getting sensations that are going into its brain. But over weeks and months, typically months, it'll start to realize things about the world. It'll start to realize that there's these sensations that are distinct colors. It might sense that the house, the room that it's in most of the time has a yellow color in it. So its brain may start to recognize the sensation of yellowness. And that sensation of yellowness will go in a particular area of the brain. Okay? And that probably is tied to a little bit of the physiology of our brain, because in past discussions, we've talked about how, for example, with vision, the retinal matrix goes through the optic nerve. It passes through an optic chiasma. There are many structures, intermediate structures that it passes through. Ultimately, it reaches to the rear of our brain, to the occipital lobe. And by that time, a lot of the visual field has been kind of broken down in very simple ways. And one of the ways that it's broken down is in terms of colors, okay? Because our retina basically has typically most people's retinas have 3 color receptors. And actually it's more complicated than that. They have 3 color receptors. Some people have 2 color receptors. Those are the people who are usually colorblind and single because they're missing 1 color receptor. Some people actually are missing 2 color receptors, in which case they'll have monochromatic vision. And some people actually have 4 color receptors, which give them supersensory perception. But all of these sensations from the color receptors go into the back of the brain. And so if this child is seeing yellow all of the time because they're living in a yellow room, their baby room is color painted yellow. The activation of the neurons that correspond to green and red, I believe green and red combined in a certain ratio will give you yellow. And so the concept of yellowness comes into play when both clusters of green neurons and clusters of red neurons both activate within a certain percentage of each other. And that gives us a sensation of yellow. Right? And after a baby sees that over and over and over for weeks and months, the concept of yellowness comes to being in their mind. They don't understand it as a word yellow. They won't understand it as a color. They'll only understand it as a sensation. But as they grow older and as they learn language, and their parent will teach them, this is yellow banana, then they'll associate the sensation of yellowness, because when they see that banana, the sensation of that yellow region is activating, right? And they don't have a region for banana yet because they're just learning the concept of banana. But over time, they'll form another region in their brain, which corresponds to banananess, okay? So this region of yellowness and banananess, you might say, those two together will, over time, start to activate, and then they'll understand the concept of a yellow banana. And then if their parent is also teaching them a language, the parent is typically using words. So they're saying the word banana. So the child is hearing this audio version, banana, right? And that creates another region. This would be much more complicated, but it would be a region of an audio phoneme. It wouldn't be a phoneme, it'll be an audio spectrogram of the sound of the word banana, right? And depending on the language you're in, it'll sound differently. And it'll encode in the temporal lobes, usually the left and right temporal lobes. But if the child is also taught written language, then yellow would have a script, associated with it, YELLOW or whatever script, whatever language script that you're learning, etc. The concept of color blindness actually is more perceptual. It's not the brain. So a person who has color blindness, their brain, we believe their brain never forms. So if a person is, let's say they're green colorblind, so they have very few green color receptors, right? So they pick up red and blue, and they pick up the green kind of look like brown to them. So what will happen is that they won't have a recognizable green sensation. What they'll have is a sensation of brownness, okay? And things that we would call green, they'll see as shades of brown, or they might see as shades of more blue. If something is more bluish green, they'll see a shade of bluish brown or more blue. So, question around color blindness is that it's more physiological rather than, it prevents the brain from forming the concept of that color, encoding that color. Although in the language area, the person will still encode the language concept of green, even though they can't see green.
Thank you. Next up is going to be Claudio, Kevin, Linda, Ed, Vanessa, and Laura. Claudio.
Well, thank you, Shrikant and Sanjay. Yeah, early on you mentioned that the brain state is a collective pattern of a group of neurons. So I was wondering, is that the same for all mammals, reptiles, and fish? If so, is that strong evidence that all life forms, including humans, are descendants of the same single life form billions of years ago?
So you're asking two different questions. One is the evolutionary track that all life on Earth that we believe has taken prior to fish, single-celled organisms in probably the ocean, to fish, to larger reptiles and mammals, etc. So they all have neurons to some level. The size of their neural clusters are different. So fish also have a brain, but their brains are very, very small compared to a human brain. Lizards have larger brains. Smaller mammals and larger mammals, their brain size gets larger, and also the regions of individual brains also get larger as you, in terms of evolutionary time, as you get further along, in terms of the evolutionary status of an organism, of an animal. The concept of whether we all evolved from a single state of consciousness, that you're presupposing that consciousness comes from a single neuron, okay? Or that consciousness comes from I'm trying to think of how to say this. I mean, we don't know exactly where consciousness comes from. We don't have an answer today. Okay, that's something that's being researched that probably will take a few more decades at least. And whether there's a link, I mean, in terms of the lower forms of life, insects and fish and things like that, they probably don't have a level of consciousness as what we would describe. They might have a sense of awareness. Well, fish seem to have a sense of awareness, but insects don't seem to have even that. But the sense of, and again, consciousness is a multi-dimensional word. It actually has so many meanings that it's very difficult to talk about. If we're talking about consciousness as in self-awareness, that we don't believe lower life forms have that.
Thank you. Next up is going to be Kevin. Kevin, go ahead.
Thank you, Sanjay. My question is regarding to neuron and brain cell self. Let's see, there's a hierarchy between those neurons. For example, some neurons control on master other brain cells or neurons. Or some neurons are more important than other neurons. Is my statement correct? And thank you.
Okay, so this question is actually much more complicated than we can get into, but I'll try to answer it in a simple way. Basically, what you're asking is the importance of neurons, or the importance of a neuron being activated, and the picture is not as simple as what I laid out. I laid out a presentation where we have a set of neurons which are either on or off, or whether they have multiple states of activation. But what actually happened is that in various parts of our brain, for example, the basal ganglia, the basal ganglia is full of many, again, millions, hundreds of millions of neurons. And a lot of those neurons are responsible for turning off regions of our brain, turning off other regions of our brain. So for example, when we're in a very crowded restaurant, okay, if our brain was very simple, we would be hearing all of the ambient noise around us, and we would not be able to concentrate. We would not be able to tune out that noise. But one of the things that these clusters of neurons in the basal ganglia do is they allow our brain to automatically, so we don't have to think about it, automatically attenuates it. It weakens the signal of a lot of the audio of the distracting sound from the ambient environment around us. Okay? And our brain does that automatically because we learn to do that. One of the things that and not just learn to do that, what happens is our frontal lobe is one of the main things that keeps our awareness. Okay, so when we're aware of our environment, when we're sitting at a restaurant and we're talking to somebody right at our table, and we're aware of the conversation, our brain knows there's a person there. So our brain knows it has to tune out the surrounding noise, and it knows it has to pay attention to only the voice of that one person in front of us. Right, so the brain activates many other parts of the brain to turn off many parts of our brain. So it turns off certain aspects of our hearing, okay, so that we're not able to hear as well all of that ambient noise in the restaurant. And it keeps on the part of our brain which focuses on the voice of the person in front of us. Okay, so it gets a lot more complicated. And this is just one example. If we're in an environment where we might have that, and the person is speaking in a language that happens to be new to us, like maybe that person is not a native speaker of English and they have a strong French accent, right? So the fact that we have to listen to them may employ other parts of our brain. So we may have to do not only tuning out ambient noise, we may have to bring in other parts of our brain, which adds creativity to our interpretation of spoken language. So it gets very, very complicated. I don't think we can go into it, but it's a wonderful question. But the quick answer is no.
Thank you. Next up is going to be Linda, followed by Ed. Linda.
Sanjay, thank you. Your presentations are always fascinating and thought-provoking. I'm thinking of what you said in relationship to the video about functional MRI. When someone is asked a question and they're having a functional MRI being done at the same time, does the brain first work on the question, the background to the question, and then go to a yes, no place? Or is there no way of finding the yes, no on the functional MRI? If that's possible, we have a new lie detector that's better than what we've been using.
Okay, wonderful question. So first, let me say that we're actually going to, in the talk later, I'm going to go into that. I have an example that we're going to actually walk through a scan of a person in a situation like that. We're going to walk through and we're going to try to figure out what's going on. So it is possible, but at the same time, it's not possible in the sense of a lie detector. And it's also not possible in the sense that functional MRIs have limitations. So there's a limitation in technology that we have today, and there's a limitation in the amount of information that we can collect using this technology today. Both of these limitations today don't allow us to do that. Theoretically, okay, if the way we understand the brain today is accurate, and it seems to be, so far, but if we can extrapolate from that, yeah, we probably will be able to have very, very reliable, I don't want to say lie detectors because lies are a complex thought. We might say thought detectors, like, is a person thinking about a blue banana? Mm-hmm. Okay, we might be able to pick that up, but not whether a person is lying about seeing a banana that might be blue or might be orange. Okay, see, that's a more confusing concept, right? A person lying about whether they might be seeing a blue banana or an orange banana. That thought, that's a single thought, right? That thought is much more complicated. We probably will never be able to discern, well, I don't want to say never, but it will probably take us many decades, maybe 10, 20, 50 decades to get to that point. But the level you're talking about detecting a person's thought, we actually can do that to some extent. The first video that I posted, if everybody saw it, you will see that we are able to actually see a thought. Not only see a thought, we're able to figure out what a person is thinking in a simple sense. Okay? If you haven't seen this video, I would encourage you highly to go back and look at the first video. It's in the description for tonight's talk. It's about 22 minutes long. It's a phenomenal video. It'll change your perception of what we're aware of as far as a lot of the activity in our mind. But in that video, what they basically describe is that we are able to, so if a person is within an fMRI machine, okay, and they are asked to think of something that they won't tell the researcher. Or let's put it another way. They're flashed a picture of something, okay, and they don't say it. The researcher doesn't know what the picture is of. The researcher is only able to look at an fMRI image sequence of their brain. And using that, and using some additional software, the researcher is able to figure out exactly what the person saw. And if they see multiple images, for example, if they see a hammer and a house, okay, the researcher can tell they're looking at a hammer and a house. Okay? So the level that we're able to understand right now, what types of sub-elements of thoughts, okay, again, these are not full thoughts, these are sub-elements of thoughts, we're able to figure out and understand exactly, in most people, even people who were not part of the study, they took other people who were not part of the original study, and they had them go through the system, and they found that they still could detect with a very, very high precision what those people were thinking. And I'm going to go into that a little bit later, what that means. But Linda, quickly, lies we cannot tell yet. We probably will not be able to for a long time. But as far as understanding what concepts, the simple concepts and crisp concepts, you might say, discrete concepts, we can pick up. We will be able to pick up with more and more resolution as time goes on.
Thank you. Next up is going to be Ed. Very, very quick, Linda. There are 7 people waiting.
Okay. Part of why I asked, they said when someone was shown something like a hammer, the brain would light up not only in terms of a picture, but the actions that were involved with the item. So if we're able to even separate the action and the picture, that's why I asked the question, but I understand what you're saying. Thanks, Sanjay.
Right.
Thank you, Linda. Next up is going to be Ed, Vanessa, Laura, Ian, Kristen, and Waldo. Ed.
Yes, thanks, Sanjay. A great presentation, as I would expect from you, and great. I just wonder if it could discuss the alternate realities that can be brought about by the use of so many different psychoactive medications that can literally change people's personalities. And I've seen how this has been, done, and alternate realities, especially for the elderly and people with deteriorating function, brain function, loss of cognitive function. There are so many ways that alternate realities can be developed to improve the quality of life. Of course, it would have its sinister aspects too, but I think that's something that should be discussed, how these drugs so affect the synapses and create a totally different, almost depersonalization, and it can be used for good or for evil.
Yeah, I mean, psychotropic or neuromodulating drugs, medication, and they can have a very significant impact on a person's brain. They can alter their states of reality. They can alter the states of consciousness. They can distort. They can make them act in ways that they would not normally act. I mean, that's an area that is. I mean, it's separate from what we're talking about tonight, but it's very, very complicated because when we're talking about thoughts, if let's say somebody is on a hallucinogenic, which is an example of a neurotropic drug, the activation level that is normally required for them to have a thought may be lowered. So spontaneously or more easily, they may have certain types of thoughts appear in their mind than would happen in a normal state. For example, in a normal state, you probably will not have fear from the wall, near you, right? Because that wall is not going to attack you. But in that type of state, if you're on a hallucinogenic, you may actually have a sensation of the wall moving, or you may have a sensation of the wall being, getting closer and closer and surrounding you. So, these thoughts and emotions can be very, very vivid and as real as we experience every day.
Wonderful. Thank you. Next up is going to be Vanessa. Vanessa, what's your question?
Okay, sorry, this may seem simple, but silly, but I wanted to give a concrete, simple example. If something's been like misfiled or mischaracterized, like stored in the person's brain, such as if you've actually never seen an eggplant, you may just know it's edible. You may picture a plant that has fruit or edible part like an actual egg. And let's say that was currently coded alphanumeric, I'm just making this up, as F3R17, but the correct corresponding code is actually 4GBB9. Will the brain make adjustments? And is there a limitation, let's say once you've entered into your 20s, where maybe it'll get stored somewhat correctly, but it'll never get to the true place where it would belong in someone that knew what an eggplant was from, let's say, age 7 on, like the color and what it looks like. If that makes any sense.
Yeah. So if a person has never seen an eggplant before, and I'm assuming that they know what the purple color is, they'll be able to recognize that color as distinct, even if they can't. They'll be able to.
They don't even know. They just know it's edible. That's edible. They're picturing a plant with an egg on it. That's.
Oh, you're saying they've never seen an eggplant, but they've only heard the word eggplant? Is that what you're
Correct.
asking?
They just know it's edible. It's a food. You can eat it.
But they've never seen one, actually. Is that part of your
Correct.
question?
They're imagining a plant with an egg. Eggplant. Well, let's see. They're forming their own idea of what it might be.
Right. So I mean that actually happens in childhood, because when a child, when a child is taught, things that they've never seen, Santa Claus, for example, right? Because most children have not seen Santa Claus. They're taught that it's a man, He's got a big belly. He's got a big, long white beard, things like that. So they put together the concept using other things that they already understand. So if your question is that how would they encode an eggplant in their mind if they haven't ever seen an eggplant? Well, seeing has to do with the visual system, right? And if they're hearing the word, they're hearing the word eggplant, so they've already heard that word described to them many, many times. And so in a sense, they've encoded the sound of the word eggplant. They might have encoded the spelling, if they know how to spell it. They've encoded these concepts in other areas of their brain, but they have never encoded a visual reference to it. So to them, the closest visual reference, because eggplant has, the concept of eggplant has two words, egg and plant, the closest visual reference to them might actually be an egg. An egg could be any egg in their mind. It doesn't have to be a chicken's egg. It can be an ostrich egg or a robin's egg, and a plant, right? So yeah, that can be. It's one way. It's possible. It's hard to say because every person's different and it depends on their personal experiences. But it's plausible that they would encode something they haven't seen using familiar concepts they already know.
Thank you, Laura. What's your question?
Okay, Sanjay, I want to go back to colors, okay? I want to think about primary colors, red, blue, and yellow. So those are your, let's say, your three major neurons. And you want to look about, when you were talking about the not as intense neurons. So let's say you have green, okay? But where does that come from? It can be yellow and blue can make green. So, those two neurons could make the green, right?
Laura, can you just ask it as a question? So Sanjay can respond to it as in actually how Sanjay, colors are.
Right. Something like that. There are primary colors. So there are three sort of strong things. And those mixtures of those colors make secondary colors like green, orange, and purple. And I'm not sure how that works with the neurons. Is that something that comes out of that process, or is that something after the fact that you learn because you put them together, tangible things that you work with and you create and then you define?
Okay, so yeah. Yeah, no, I understand what you're asking. So vision is extremely complicated, and there are two primary areas of three areas of vision that we're not going to go into detail. But what happens in the retina itself is one area. What happens prior to the occipital lobe in the medial regions of the brain? There's a lot of processing and moving around of the spatial information, and then ultimately what happens in our occipital lobe. So the retina itself is a very, very complex organ. First thing I want to clarify is that when you're talking about color space, the primary color is actually red, green, and blue. And the reason why we consider those primary colors is because we have color detectors. We have cones in our eye, which correspond to a range of green wavelength, a range of red wavelength, and a range of blue wavelengths. Okay? And in each person, the sensitivity of each of those cones vary, as well as the distribution of those cones, the density of those cones vary. So for example, in most mammals, there are more blue cones in the lower half, in the upper half of our retina, in the lower half of our retina, because the sky tends to be blue, and we need to see blue in our environment. There are fewer blue cones in our upper half of our retina. So there's many, many complex things in our retina itself, which affect how we see the sensitivity that we have to different colors. So when you're talking about the color yellow, which is a combination of red and green, okay? So for us to see yellow, the blue cones would have to be almost inactive. Okay? And just before I go there are cones in our eye, in our retina. There are rods which detect intensity. And then there are neurons, there are neuronal cells which connect to the rods and cones. And it's not a one-to-one, it's a very, very complex relationship. So for example, you may have one neuron that connects to half green and half red cells. So that neuron, in a sense, might become like you might say, a yellow neuron, because it's picking up half green and half red. There might be another neuron that connects only to red cells, to the red cones, and that neuron will be a red detector. So there's multiple layers of abstraction going on here within the retina itself. Okay? And then those neurons that have color specificity, these neurons and these rods and cones form a matrix, a literal two-dimensional matrix, which corresponds to the visual field that we see. So when we see the retina is pretty much spaced all around our field of view, right? And so those retinas send, not retinas, those rods and cones send the signal to the corresponding neurons. Those neurons aggregate and send the corresponding signals to the optic nerves, to the central region of our brain, the medial initial processing of our brain. A lot of processing that happens that I'm not going to go into it. And then it reaches the occipital lobe. And the occipital lobe is actually where we try to recognize what we're seeing. Okay, it doesn't deal with color sense. It doesn't deal with color processing. The color processing happens in our retina, but our brain at that point is getting a sensation of yellowness or blueness or greenness or orangeness or whatever color, or a zigzag or a zebra pattern, or of different visual sensations, and there are further processing that happen there. I don't think we can go too much into this because this is a little bit off topic, but hopefully this answered what you wanted to understand. We can go into this later in future talk.
Next up is Ian. Ian, go ahead.
Yeah. Okay. Is this clear?
Yes.
Yeah.
Okay. From Sanjay's presentation, of course, a thought consists of myriad loci, positions of neurological activity dispersed along countless brain areas and the like. And my question is basically, again, following from Sanjay's overview and presentation, that basically a conscious thought, so to speak, would seem to be, can we say that conscious thought is roughly, this is very, very rough sketch, basically the prefrontal cortex sampling this vast ensemble of neurological activity, sampling it to not pick up the entire global ensemble, but basically zeroing in on particular snapshots that seem particularly pertinent for capturing, shall we say, the essence or the summary of it all, and also moreover as a springboard, a stimulus for further action. The idea being, okay, the thought has this myriad amount of neurological activity. Basically, what's the next step, so to speak? How is it going to stimulate action on the part of the organism and the individual? And basically, the question is what we perceive as conscious thought basically this snapshot of this global activity that's meant an action stimulus?
Very good question. Yeah. And so the quick answer is yes, more or less. But let me just delve into some things that you brought up, because I think it's important to differentiate. In the examples I gave earlier, I talked about the frontal lobe sampling, and I use that word, but delving into a little bit more detail, we're not sure if it's necessarily sampling that the frontal lobe does, or it may be that the various regions of the brain send a status to the frontal lobe, or it may be a combination of the two. It seems to be a combination. It seems to be that the frontal lobe every once in a while is monitoring the state of various parts of our brain. And it's always keeping so the concept of awareness, when we say we're aware, like we have a awareness of our presence in the world, right? That happens because our brain is always sampling, for example, our visual system to get a sense of what happened. All of a sudden, am I in a different room now than I was 20 minutes ago? Or if a siren starts to blast, by my window, right? Then that's a very different stimuli. And our conscious mind, our prefrontal mind, will automatically be, it might be that the auditory area notifies the frontal area, or it might be the other way, that the frontal area is regularly monitoring, which might be a combination. So I don't think it's important necessarily to focus on which one it is, whether it's a monitoring or whether it's an updating, whether it's active or passive, or whether it's both. The fact is that the prefrontal area, the frontal lobe itself, and probably most likely what seems to be is the prefrontal area, is the seat of consciousness, or is a seat where our general awareness happens. And it conglomerates the statuses, you can say, of various parts of our brain at any given time, and builds a state of the overall brain and the overall body, or overall being. Right? It builds a state of where we are in the universe, in the world. I'm sitting on a chair right now. I'm in front of a computer right now. I'm speaking. Right? These are concepts that my frontal lobe is aware of, because it can sense that my motor cortex is causing my mouth to move and causing my hands to move. My frontal lobe is aware of that. My frontal lobe is aware that my eye is picking up visual stimuli of a Zoom session. Okay, it's aware that there's a light in front of me. Right, it's aware of all of these things around me, and it integrates all of those concepts into a consciousness, awareness of the world around me. So yeah, it is. I mean, and there may be more depth and detail in that, but at a very rough general level, that's kind of where what it is. And at the same time, the complexity of the thoughts and the ideas that our frontal lobe can deal with, I believe, and the scientists that I've talked to at least also believe that language may be a very important element of that, that if you have language, you actually get a additional ability to conceptualize even more complicated ideas. And the last part of your question, Ian, is that after you have these thoughts in your mind, from that, the executive function within the frontal lobe can actually then take a course on what's the next step, what's the future action to do based on the current state of the world and possibly the emotional state. For example, if the emotional state is you recognize it, and for example, let's say that your mind also picks up sensation that you're hungry. It may also pick up sensations from your stomach, right? Then it might come up with a future action that in whatever time I'm going to eat lunch, in the future, something like that. So it comes up with a course of action for the future.
Thank you. So we'll take two last questions. We're running a little bit late, so let's do two quick questions and answers, and then we can go to the breakout rooms. So it's going to be Kristen, followed by Waldo. Kristen.
Can you hear me now?
Yes.
Okay. Hi, Sanjay. Thank you so much for the presentation. I came a little bit late. I was actually coming in from a Philosophy of Mind other meetup group, but I heard most of it, and thank you so much. I just was maybe it's kind of a question that's just to kind of clarify the position, kind of like a would you agree or would you not agree with the following? And it's this. There's a difference between approaching almost perfect accuracy with the ability to say, this is what this person is thinking right now. That's different from, we have in fact not only identified what a thought is in the brain and what it consists in, but also it's not only its location, it's what makes it up, et cetera, right? Would you agree or not agree with that?
I missed the question part of that. You said.
So what I'm saying is, I'm fairly familiar with the fMRI and machine learning kind of stuff. So what we're talking about is basically the pattern recognition of these patterns of activation gets so good that essentially, like, what's happening is that in fact, the machine learns that there's 24,000 ways to distinguish the thought of a cat versus the thought of a dog, right? Right.
Okay.
Right.
What's your question? Can you go ahead and formulate a question?
My point is this: that increasing accuracy of the ability to say what thought a person is having is different from saying we have in fact located or meaningfully said something about the location, nature, consistency, and makeup of thoughts within the brain. Do you not agree with that? That those are two different things?
Right. So I think that those two have a little bit of correlation right now. I don't know if we can say that they have complete correlation or that they will have complete correlation. I mean, this is a complex question. So I'm trying to explain it in a way that everyone else will also understand it. So basically, the ability, the certainty and precision with which we can discern, and you're talking specifically about the first video. So those who didn't watch that video may not understand what we're talking about, but the precision and certainty with which we know exactly a thought that is in a specific mind, whether that correlates to, or how strongly that correlates to the neural state in that person, is a, you might say, one-to-one or a complete strong correlation. I don't think it is right now. Also, this is a very, very difficult question. It's a very complicated question because there's so many, I'm trying to figure out how to simplify to answer it. I mean, there are issues around the detection of how fMRIs work. There are issues around variation within people. There are issues around the time sensitivity of fMRIs, and as well as the resolution, spatial resolution. There are many, and that's only on the fMRI side. When we get into the neural network side of it, there are many, many layers of complexity. So all of this put together, right now, this video is a proof in concept, you might say, that theoretically we can detect in very gross, rough terms, simple neural concepts that we hold in a mind. But they're only as good as the data that was collected for that neural network. Okay, so if you take those same people who were in the study and you take them 20 years later, their brain has probably changed significantly in those 20 years, and probably that same neural network will not do what it did originally. I don't know if that's going to helping to answer the way I see this. It's not as simple as the video showed it. It's actually more complicated than that. The video simplified it. It was a wonderful video. And the study, and it's not just that study, there are actually multiple studies that have repeated that work. So it's not just a one time. This is pretty well established already in the neurosciences that we can kind of tell to a very crude level what a person is thinking of if we have a neural network which has been trained with those types of thoughts. But it's still not as simple as saying that system is always going to work with anyone or even those same people in the future.
Thank you. Last question is from Waldo, and then we're going to do some breakout rooms. Waldo. Waldo, where is Waldo? Waldo, you need to unmute yourself. Okay, let's go ahead and do breakout rooms. Okay, Waldo says his background is too. Anne, you had a question? Okay, all right. So folks, we're going to do breakout rooms for, let me see. Okay, Waldo has this question. It says, for the complexity of thoughts mentioned in the presentation, does the brain limit its outputs to conserve energy? How may this vary for a person who may feel threatened?
So, yeah, I mean, in previous talks, we've talked about energy and how the brain and body is managing energy all the time. That's a very fundamental aspect of our biology, of our survival. So yes, the brain is in one sense, it is managing energy, but not at the level of thoughts. It doesn't seem to be getting to that. I mean, a single thought is not using that much energy that the brain would need to manage it. And if the person is feeling threatened, so I'm assuming you're talking about emotion, for example, a fearful emotion. And probably the reason why you're asking this question, if I can read into it, is that when people get afraid, their sympathetic nervous system get activated, their heart rate goes up, they may start sweating, they may become very, they may, the phrase fight or flight, they might start to go into this fight or flight mode of preparedness and reactivity maybe. But those aspects would use more energy. Your heart rate increasing would use more energy, right? And your brain, in one sense, may operate slightly faster because it has to process more stimulate, wants to not miss anything, might process more quickly. But what the brain does in a situation like that, so the example, if I could take your example and push it further, is let's say the person has been activated into a fight or flight mode where they are fearful for their life now, okay? So their body has been activated, it's using more energy. Their brain has been activated also because it has to become hyper vigilant of its surroundings, right? The person's surroundings. But one of the things that the brain does automatically in that situation, it actually shuts off the blood supply to other parts of the brain and the body, which is not necessary. So the prefrontal cortex is one of those areas, or the frontal lobe is one of those areas that gets reduced blood flow. Our brain actually turns off that region, or the thinking part of our brain. So when you're in a fight or flight situation, you have less ability to think. And that in a sense may, probably does, I don't know exactly if it will, but it probably will reduce the energy usage of your brain because energy comes directly from blood flow. Glucose molecules are what drives all neurological activity. So if you reduce the blood flow, yes, the energy management aspect of the brain would kick in there too.
Wonderful, folks. Thank you. So folks, we're going to do breakout rooms only for 10 minutes. So you can talk to each other and then we can come back and then we will wrap up the second part of the presentation. Starting,
Shika, I just had a few more slides after that I wanted to cover, but okay. Yeah, it won't take too long.
Okay, so think of more questions that you will have, so you can come back with your questions. There'll be 2 slides and then you can ask further questions. Starting the breakout rooms now, they're going to last only for 10 minutes. Welcome back, folks. Welcome back. So we're going to now continue. I think Sanjay has 2 more slides, after which you can ask more questions. Sanjay. Sanjay, you're on mute. Go ahead.
I was muted, yeah. Sorry about that. So yeah, it's more than 2 slides, but these are very quick. We can go through them pretty rapidly there. So let me just get that. Okay, so what we're going to look at is some of you may have seen the second or third video. If you did, then we're just going to do a very quick walkthrough. So a thought in slow motion. So here, this is a video where there was a study in which they had 2 different situations. The first situation was a person was laying in an fMRI scanner, and they were given a word, a specific word, okay? And they were asked to repeat the same word after they heard it. And on the top of the slide, you see the audio waveform, which is, you'll see 2 different squiggly areas. The left side is the word is humid in this case. The word was spoken by somebody else. It came out of a speaker. And the right side is when that person who was part of the study, they repeated the same word using their mouth. So we see the word humid. They hear the word humid, and then we see them speaking the word humid. And then at the bottom, you'll see 3 different colors. You see yellow, which corresponds to stimulus perception, which means you might say that's the hearing part of their brain, right? The sensory part of their brain. And then in red, we'll see the word evaluation, which is that they're trying to figure out exactly what word they're listening to, and the fact that they're listening to audio, and the fact that they're listening to a language. And then in blue, you'll see that they're starting to prepare their mouth and their body and their mind to say something. And then they actually do the same, they execute the same. And so we're going to flip through these in a little bit of a rapid succession. The video itself is I think 7 or 8 seconds, so it goes very quickly. But here we're going to go through a little bit slower motion. So I want to explain exactly what the previous slides that we talked about, the presentation. This will demonstrate exactly what is happening in an actual live brain in a situation. So first slide is when they start to hear the sound for the word humid. And what you'll see is three regions: a yellow, a tiny blue region, and a small red region. What this means is that the person has large area in yellow, which means that they're hearing a lot of sound. So their audio sensory perception is fully activated, strongly activated. And there's a small region in red. That region in red might be that they become conscious of being able to hear a sound. Okay, so this is when we're talking about, Ian asked the question earlier, of consciousness, okay? That this might be the conscious aspect, that they become conscious of hearing a sound, and they have to interpret that sound. And then there's a tiny, tiny blue area, which might mean, because this person was given instructions ahead of time, right? As part of the study, they were told, you'll hear a word, and then you have to repeat the word. So that blue area might be that person preparing. They might be preparing themselves to say the word. And initially, it's a very, very small preparation. It's a very small region. And actually, for those who know the biology of the brain, that area is actually in the facial area, okay, where the blue is. So it may correspond to their mouth, or it may correspond to their ear, but it does correspond to the facial area. And then just a microsecond, a few milliseconds later, after the word human is almost completely spoken, you'll see that the yellow region, the hearing part of it, is very strong. And by that time, they probably have begun to grasp. And then the red areas, there are many, many areas in red, which are mostly in the prefrontal area. So the left part of this brain is the prefrontal area. That's the thinking part of the brain. And so they're doing a lot of processing in the red area to try to understand what the word means, right? And then there's a little red area in the center, which corresponds to the language area, the Wernicke area. So they may be trying to process exactly what that word means, or the concept of the word. This is the language center. And then you'll see the blue is much, much stronger. So they're probably getting very, very ready, because by this time they're realizing that they've heard almost the entire word. So now they have to repeat the word. So they may be energizing, and you'll see the activated area is higher up here. So this corresponds to other areas of their musculature. So they may be getting ready to activate their vocal cords or their diaphragm, because to speak, you have to move your diaphragm, your lungs. And so they may be energizing, getting prepared to activate specific muscle groups in their body to start to talk. And then a few milliseconds later, you see the yellow region is gone. So they're no longer hearing anything. Now the red area understands exactly what to say. It's directing actions on what to do. And the blue area is fully ready to begin speaking. And now a tiny blue area also began. So this second on the right side, so if you look at the previous and this, okay, there's only one little area. I'm flipping back and forth, right? So there's only one little area between these two that is activating. And there's one, and the red area is no longer activated, okay? The red area in the center is disappearing, okay? And the blue area is appearing. So what this means is the Wernicke area is no longer processing language, because the word has already been spoken, right? They've already heard it. They've already finished processing the language. So that central red area is gone. And that new blue area is probably this area of the brain, the right side, right top part of the brain is the parietal area. And this is, this has to do with recognizing your body. So they may be recognizing, the person may be recognizing that their ear is acting, because you see that the audio waveform is starting to, they're just starting to enunciate the initial sounds. So this may be that they're starting to recognize that their mouth is moving. We're not sure exactly what, but it may have to do with either motion of their mouth, or it may be that something their body's changing. And then you see the yellow area appear again. And this is because what's happening is that as they start to say the word, they're actually hearing their own sound, their own voice that's heard by their own ears. And that's why the yellow area has come up again, because they're hearing their own voice. And the blue area is much, much deeper here, because they're in the middle of enunciating, which is using a lot more of their brain. And then as they go further, the red area diminishes. The red area eventually disappears, because they no longer have to think about it. They've already thought as much as they need to. All they have to do now is enunciate the word, which they're still doing. And the yellow area is still there. They're still hearing it. And then as they go through, eventually, it's done. So they're done with that aspect. So this is an example where they hear a sound, right? So that's one part of it. Then they have to think about what that sound meant. So those are the red areas. And then they have to take an action. And that action part, so Ian asked about, that when you have a thought, that may lead to a future action. So in this case, that did lead to future action because the person was told to repeat the word. So this is a cycle. So we just have one more of these, which is quick. So in this case, there's a slight difference. They were asked to listen to a word, and they were asked to give it the antonym of that word, the opposite of the word. In this case, the person heard the word humid, and they chose to repeat the word dry. So the mental processing is a lot more complicated in this sense, because not only do you have to hear the word and recognize it, but you have to hear the word, you have to recognize what the word means, you have to go back into your memory and find an antonym, which is actually very complicated and it takes a lot more time. That's the reason why the gap between these two activities is much longer. The gap between the time scale on both of these studies was the same. So that's why this gap is longer, because it took the person, literally took them much longer to actually think of an antonym. That's why they started speaking much later. So I'm going to quickly go through this. So initially, they have the sensation of hearing, which is yellow. And then the bottom two red dots have to do with the Wernicke area, which is again recognition of, this is the language center of the brain, which is recognizing the language. And then the top red area is most likely recognizing that they're becoming aware of hearing something, right? Recognizing that they're the presence in the world. Then after they've heard the word, now you'll see lots and lots of red in the prefrontal area. That's because, actually this is in the frontal lobe, it's not necessarily the prefrontal area. There are lots of red because this takes a lot of recognizing, a lot of memory, right? A lot of processing. And you'll see the Wernicke area on the bottom, the red, is still active because they're trying to figure out what is an antonym, what is the opposite of this word. And they're going through their memory, trying to think of what words are the opposite of humid. And they may be coming with multiple words. So what they might be doing is choosing which is the best word. They might have come up with two words. They might also be deciding between which of these two words. Then they're no longer thinking as much because they've already decided on the word. Now what they have to do, they have to take action, right? So there's still some red activation in the frontal lobe, but the blue area, the somatosensory part of their brain is now gearing up to begin to speak. So again, they speak. And as soon as they start to speak, again, they hear their own voice, which is the yellow. Then after they're done speaking, the blue area has subsided, and then gradually it gets weaker. And then they're saying the last few sounds of the word dry. And you see this takes a lot more time, and it eventually tapers off. Now, one of the things I want to explain here is that going back here, now these images give the presentation as if when you're taking an image using fMRI, that the scan is clean and crisp. Okay? This is actually not how it is. These images are actually very, very, they've been processed and they've been cleaned up a lot for the study. And the processing and the cleaning up that they did is based on other knowledge that we have about how the brain functions. So they basically, in a typical MRI, they would see activation in almost every other part of the brain to some level, not to a high level. But what you'll see is that the levels that they've highlighted here are the stronger activated areas of the brain. And the other areas that don't have any colors in them probably do have some bit of activation, but they're below a certain threshold or they're not distinct. Also, these images more than likely are averages over time. So for example, this person was probably told to repeat this multiple times. They were probably asked to do this exact same experiment 5 or 6 times. And so each of these slides is an averaging of the images from all of those 5 or 6 runs. And then this is the best case data that's presented. And also what they've done is the color coding. The fMRI would not have the color coding that's artificially colored to help us to understand. What fMRI would show is that they would show all of these regions activated, but because they know, and in this, I don't remember if in this study they also used a artificial intelligence tool to help them discriminate between the specific features of the brain. That may have helped them to colorize it, or they may not. They may have relied on their own intuition. But taking these types of measurements and doing these studies is actually much more complicated. The data that oftentimes you'll see is it's simplified to such a level, but the raw data is very complex. It's very difficult to interpret. There are multiple levels of pre-processing that's required to do all of that. So I'm going to stop here, and then we can, if we want to do questions, answers, or we can do, actually, I'm going to stop here. I had some more things on the network aspect of the brain, but we can do that later. It's getting late.
Yes. So thank you, Sanjay. Let's see. So folks, now it's time for, you can either talk about, you can ask a question, or you can talk about your takeaway very briefly, in less than a minute. What did you get? William, you had a question?
Yeah, I had a question on those last two slides that were put up. As I understand, the fMRI works on the basis of blood flow to various parts of the brain. Was it taken into account, the time lag between what actually happens and the blood flow to that particular part of the brain where the neurons are actually firing?
So yeah, so that's a good question. So fMRI actually uses many, there's several different types of fMRI. One of the types that you're talking about is bolded. That's the most common ones. It actually looks at blood oxygenation levels with that in neurons. It's actually not measuring the blood flow necessarily. What it's measuring is it's measuring the oxygenation. So in a sense, it can measure blood flow, but that's some of the information that gets taken out from the measurements. So the net data that comes out of the post-processing in an fMRI system is it removes the moving blood, you might say, and it only gives you the information about specific, it's not neurons necessarily, it's regions of the brain, it's voxels, three-dimensional, you might say three-dimensional pixels, they're volumetric pixels, that's what a voxel is. So they're looking at the amount of metabolic processing of oxygen in the brain. And this is one of the reasons why fMRI cannot detect axons and signal flow. It can only detect, this is one of the limitations of our technologies today. It can only detect neuron activation. But there is a slight gap, but the gap is not large enough that it would, in this type of experiment, the gap would not cause any significant degradation of the quality of data. What would happen is there would be a consistent and fixed shift in time, which would be on the order of probably several hundred, very low milliseconds of shift. And if you look at audio frequency, it would be within the resolution of that they would be able to detect it. And they probably have shifted the time scale slightly anyway, because the audio waveform can be shifted in time relative to the image. So they've more than likely they've adjusted for all of those factors.
Thank you, Sanjay. Sanjay, we have got about 5 or 6 questions already. So may I request you to keep your answers relatively short? It's already around getting to about 11:08. So I wanted to make sure that we don't keep it too long. So next up is going to be Linda, Joe, Vanessa, Ian, Claudio, and Kristen. Linda.
Sanjay, in our breakout, I had the sense that there are people who have more than a small amount of skepticism about this, and also people who fear it will be misused. My question is, do you know if there are many academic centers doing this same research to check the validity of the studies that we have seen? So
the study the video that I posted, that has been replicated a few times. I don't know how often. I suspect it's not being replicated that often because the 2 of the studies I looked at were fairly well done. They were established and they seem to be not have any significant types of problems. So they do show that this is something that is real. I'm trying to think of the best way to describe it. It seems to be representing accurately what is happening in the brain, but at the same time, there's 2 parts to this that we have to understand. There is the imaging part of it, right? The fMRI part of it. And then there is the artificial intelligence part of it. The artificial intelligence part of it is something that is going to expand and grow and change drastically. And the imaging part of it also will expand and grow and get sophisticated, but there's a much longer time frame for that. If people are worried about this, if you watch the video does go into this a little bit, but this is not something where we can read people's mind today. it is not possible to read anyone's mind today. And at least I would estimate for at least another 10 years, probably longer than that. But the fact is, this right now requires an MRI, which is a huge machine. It's not a pocket machine. It's not a helmet you put onto someone's head, okay? So they have to be in an fMRI. Also, the artificial intelligence engine has to be specific to the types of information you're gathering. So there's a lot of tuning and processing that has to be done. It doesn't have to be tuned to that specific person. It does not have to be. But this was a limited demonstration that this technology is feasible. But more importantly, this was a demonstration that the way our brain works is actually not that different from person to person. Now, there is variation. So one of the things that we have to understand is that if we want to get into finer grain, and I understand that this is taking longer, but this is a very important concept when we're talking about thought, is that the resolution, the granularity of a thought, okay? Meaning, if you're talking about the level of blueness, or if you're talking about a hammer, right? The size of the hammer, the type of hammer, if it's a hammer with a claw, if it's a club, or if it's a mace, all of these things can be considered a hammer, and they will all activate a region of the brain what associates with hammerness, right? But to understand and differentiate between a hammer with a claw or a ball peen hammer or a club or a mace, you would have to have finer resolution. You would have to be able to discern within the several, million or 100 million neurons. You'd have to have much better resolution of exactly which of those neurons are active and which are not active. At that level, we cannot do today. And deep learning technology today is not sophisticated enough. It will be sophisticated enough, but today we cannot do any of that to the level that people are worried about. So I wouldn't worry about it right now, but in the future, 50 years from now, it's possible.
Next up is Joe, followed by Vanessa. Joe.
So my questions are almost like yes, no, and should be very succinct in the sense that, and I'm trying to make a link here, and I'm not sure if it's valid. So the first one is that if you were asked to repeat the word or the antonym, would that be the way system 1 and system 2 would look if you were imaging it? And does this have any relationship with the myelinated axons that you were talking about earlier?
Okay, so the first question is no. I mean, it does not have to do with system 1. Most of that work dealt with system 2. System 2 is the thinking part of the brain. System 1 is the fast-acting part of the brain. So it did not. The second question you asked with myelination, this required the activity of the myelinated neurons, but it was not imaging any myelinated. It was not imaging any of the myelination because that cannot be detected right now through MRI.
Next up is Vanessa. Vanessa, what's your question?
Okay, I thought you were going to get into this, but this was based on the video where it said, looking at the things, you could tell something was a chair. Well, what if this test subject had an adverse reaction? Let's say if the word was pickle, that was charted out and everything. And let's say they had an allergic reaction or got nauseous. So such an adverse reaction, might that be mapped in the brain differently where someone may think, oh, this is a skewed result, or this person was daydreaming, the person not knowing that person's bad experience with the pickle?
Yeah, that's an interesting question. So two ways to answer that. One is that when they do studies like this, they typically find out ahead of time things like that may cause, this basically would be error or noise in a study. So they may have a set of 100 words on a piece of paper, and they may ask the person to circle words that they have problems with, or circle words that evoke certain responses in the person. Ahead of time, they may screen the person. That's one approach that's often taken in studies. Second approach is that if they don't do that, that person, if they have what you might call adverse, it's not adverse reaction, the true sense of it, but their brain would react in a way that the neural network would not anticipate, because they would have some other activation. So in that case, the neural network would probably give an incorrect answer for that one person.
Okay, so now we're going to take last 3 questions and we'll wrap up. So Ian, what's your question?
Okay. Okay, actually had a couple brief ones. Mostly, Sanjay is focused on the usefulness of MRI for mapping, for visualizing these neurological activities. I'm wondering about the relative usefulness of something like PET scan, where PET scan detects basically glucose consumption as opposed to blood flow, a la MRI. Wondering about the usefulness of PET scan, say, by contrast, for imaging finer grained neurological activity. And I guess the quick second one was basically much application of MRI to on the cusp detection of neurological activity is held back by the fact that these devices are humongous in size. I recall reading that some neuroscience researchers suggest that theoretically these things could come down ultimately to the size of a laptop or even a smartphone in a distant future, facilitating their widespread use for these applications. Basically, does Sanjay have an idea about what technological advances would allow us to shrink MRIs down to, again, the size of maybe laptops or smartphones and the like?
Yeah, so first, I mean, talking about MRI versus PET scans. So each of these imaging technologies have advantages and disadvantages. MRIs have temporarily, they don't have high precision. Spatially, they do have high precision. And they also are not directly measuring activity in the brain. They're in direct measurement. PET scans on the opposite are almost the opposite of what MRIs have in terms of advantage and disadvantage. They do directly image metabolic activity, but their spatial resolution is much poorer, but their temporal resolution is much better. So in a sense, they're complementary technologies. And that's the reason, and because the spatial resolution, the resolution of the image of the voxel, you might say, is so much larger than MRI, they're not used as much for research like this. As far as whether we can shrink these technologies down, theoretically, yes, but it's going to be very difficult because just looking at MRIs alone. Right now, the research grade, the top type of MRI instruments, the ones used in research are running around 9 Tesla, close to 11 Tesla, which is a very, 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 very strong magnetic field. Okay? To generate that strong of a magnetic field from something the size of a cell phone would require a lot of energy. So that cell phone would need a pretty hefty battery. Okay? A battery that literally would have, you know, the amount of energy that's in, let's say, a gas tank in a car. That's the amount of energy that phone would have to have in your hand. So theoretically, it's possible, but there are a lot of practical implications. And also an MRI would have to have spatial resolution. So that 9 Tesla or 11 Tesla field, you know, if you're moving an MRI machine, works only because the person's body is fixed and stabilized within. The body is not allowed to move much. But if you are holding a cell phone in your hand is going to move that cell phone, so you're not going to get the resolution. So you have to kind of freeze the cell phone and your body relative to each other to make that work. So there's some physical constraints that are very challenging.
Okay, next up is Claudio. Claudio, go ahead.
Yes, based on the assigned readings, there are like trillions of connections in the human brain, or synapses, right? Based on the last slide that you showed us, there appears to be a lot of redundancy in the network of these connections. I'm curious, are there many, if any, synapses that are no longer traversed during the modern Homo sapiens lifetime?
So these synapses actually grow as part of the brain growth. It's not that they're there from the beginning. When a child is born, child has roughly 100 billion neurons that it grows within the first year of life, after birth. And those neurons are not very much interconnected. The interconnections actually occur. Some of the interconnections start pre-birth, but most of the connections start after they're born. And so these synapses don't really exist. There are very few synapses that exist. I mean, if you're talking about, hundreds of billions of synapses, trillions of synapses, compared to trillions, we might have just a few 100 million synapses at the time of birth, but they start to grow. Also, the experiences that we have in life, what our brain senses is actually what causes synapses and axons and dendrites to grow or die off. There's pruning that happens all the time in our brain actively, especially during the infant years. One of the earlier talks I did, I went into that earlier in more detail. So a lot of this synaptic pruning is very critical. So actively when we're living day to day, synapses that are not used actually get removed.
Last question is from Kristen's line. Kristen.
So thanks again, Sanjay. I was part of that camp that was like registering some skepticism. Just to clarify, skepticism from my perspective is not coming from the studies themselves. This study is like one among now thousands. I mean, this is being replicated all over the place. So the science and the field of inquiry itself, for the most part, it's good replicated, inquiry. To me, the skepticism is coming from how it becomes appropriated, especially in popular treatments. I want to just kind of re-ask that original question I asked at the beginning when I asked it in a clearer way. Sanjay, would you agree that approaching kind of perfect accuracy of identifying what thought is happening inside the brain of a person at any moment is different from saying what is actually the title of this meeting, what does a thought look like within the mind? That there is actually still a categorical distinction between these two things. And it kind of gets, we are, because of the impressive achievement of ML algorithms and interpreting and distinguishing fMR data, it becomes very easy to confuse, their performance accuracy with like peering inside and like reading, oh, there's that thought exactly, there, and that's what it looks like.
Okay, so I'm not sure where, so you said you had skepticism. So I'm not sure if your skepticism is on the machine learning side or
no. It's not. It's not on the machine learning side. It's not on the. It's not in the field itself.
Yeah, I got that. Yeah.
Yeah, it's on the field itself. It's in very subtle.
Integrating the two.
What's that?
Integrating the two.
Well, no. It's basically there's a vulnerability to conflating the performative accuracy with like actually saying that is a thought there. That's what a thought looks like, right? There is a vulnerability to forgetting that what we're talking about still is a proxy measure. It's just a very impressive proxy correlation. Like it's a very, like you said, because we're using machines, it's like the most granular correlation that we've come up with, but it's still a proxy for what we mean by thought. So I'm saying the title of this thought, what does a thought look like within the mind? That is in itself kind of an overstatement of what is actually happening here.
Okay, so we're talking about granularity. So granularity always has resolution associated. Okay, so I can answer this in terms of today, it's one world, right? One view of the world. And in the future, it will be different. And far in the future, it will be very different. So the granularity will increase to the level at which neurons exist, right? So the granularity can never needs to go below the level of neurons. Or you might say that, well, theoretically, you can go down to the level of neurotransmitters, right? Each individual neurotransmitter, right? So I'm going to take this to the maximum extreme level that we can, is that if you have granularity to the level of every type of the roughly 30 neurotransmitters we have, and the combinations and permutations of activity that can happen, if you had the ability to sense at that level, okay? So you had perfect knowledge about a specific person's brain, and you fed all of that data into a very sophisticated neural network, okay? A deep learning system. And that deep learning system was trained on that specific person. Okay, that's the key part here. I'm gonna get back into that. If it's trained on that specific person, then that neural network will 100%, if the training is well enough, its training is done correctly enough, that neural network will be able to predict 100% what that specific person is thinking. But this is probably decades, if not hundreds of years away from today. This is not anywhere near today. Okay? So one of the variables here is that the variability between each person's mind and the variability within a single person's mind over time, these two things have not been dealt with. My personal feeling is that those are humongous obstacles which will put a challenge to this technique on the side of both imaging and of the machine learning. But the machine learning part actually will advance faster, because here's something that is important to understand. The machine learning part can actually learn to infer. So for example, actually this was not in the general discussion, this was in our breakout, but the machine learning right now is trying to guess. I mean, and that's what machine learning is doing, it's guessing. But if you think about it in a sense, that's what our brain also does. Our brain is guessing about the world. So it's a proxy for that in that sense also. So machine learning is trying to guess at what a certain pattern of activation in a certain region means, okay? But if you had the resolution down to all of the neurotransmitters and further, okay, and you had perfect knowledge of that region, then you would have, and you've trained on that, then you would have a perfect understanding of all of the different combinations that that region would have, and would correspond to in thought, right? In the human world, in our world of understanding. So, and that machine learning tool can be trained to look across all of the variabilities. So for example, you know, in our talk, in our breakout room, somebody talked about the word blue and how the word blue also has a meaning in psychology, right? And mood, okay? My mood is blue. So the machine learning algorithm eventually can be taught, or it probably will learn, as soon as it's sophisticated enough, it will learn to associate the concept of blue, not only with color, but with many, many other things, anything and everything that human beings associate it with. So we're talking much further down the road. We're not talking about the technology that this TED Talk described. TED Talk is a very, very simplified version of it, but it only showed that we're heading in a direction where we can start to understand just a teeny weeny bit right now, but what's happening inside a brain. Now, the title that I gave is, you know, what does a thought look like? In a sense, this is giving us not, so in this first study, they weren't looking at individual thoughts, they were looking at sub-thoughts, okay? But the title that I gave is the next step of that, beyond that. So if you take multiple objects, as well as more advanced concepts of linking them together, and that's actually, it's a very, very advanced step. It's not a simple step. We're not gonna make this step in 5 years or even 10 years. But if you make that step, then using the same technology, basically, more advanced versions of the same technology, we can, you know, quote unquote, look at thoughts. That's the premise.
Wonderful. Thank you, Sanjay. Sanjay, any closing thoughts?
No, I mean, I enjoyed this tremendously. I mean, it made me think, as usual, about areas that I hadn't looked into. But yeah, I enjoy these discussions. I think overall, though, you know, what I want people to take away from this is that, you know, and I heard that there were people concerned. This is not something to be concerned about, because there are so many limitations right now with the technology. For example, MRI machines cannot be, you know, shrunken quickly. They're physical, you know, limits of physics with how much energy you can store in a finite amount of space and the amount of energy that you may need to run it. Now, there are tremendous limits, so I wouldn't worry about this as much. But eventually, the benefits from these types of technologies will be in helping us with, not in, like, you know, people talked about lie detection. The benefits will be more in detecting, for example, mental illnesses or diseases, or it may be in for example, a fighter pilot, you know, or even driving a car. I mean, although car, you don't have to, you know, autonomous vehicles kind of remove the driver from the element. Fighter pilots probably will remove that also. But, you know, instead of a person needing to do something actively with their hands and voice and things like that, it will allow us to have more direct connection with our brain to interact with the world in more sophisticated ways. So the technology is very powerful, but it's very, very, very rudimentary. Nothing to worry about.
Wonderful. Thank you, Sanjay. So folks, let me remind you that this series, Comprehensivist Wednesdays, we do in conjunction with Greater Philadelphia Thinking Society. It's wonderful to cooperate with people from there, you know, CJ, Sanjay, you know, and others. And we cover a whole range of topics, you know, and especially connections between topics on Wednesdays. This is in conjunction, with my group, which is the 52 Living Ideas. 52 Living Ideas, we do meetups every day. I want to bring to your notice the meetups that we do on the weekends, because on weekends we try to go into very deep new areas. So for example, this weekend on Saturday at 12 o'clock, we're going to be talking about development of psychological concepts in Japan, history of development of psychological concepts in Japanese culture. At 2:30, we'll be talking about Louis Sullivan and his contribution to architecture or design or living. His idea is form follows function. And at 5 o'clock, we are going to be talking about how do infants develop a concept of consciousness? How do you go from an infant to a time where you actually see that there is something that is going on in the head of a person, that they have a notion, they have a mind, they have a consciousness, and then you yourself figuring out that you are conscious as being separate from that. So that's what we're going to be talking about. On Sunday at 2:30, we'll be talking about the trivium, which was the central educational mechanism for over 2,000 years. And we're going to be looking at the dissertation by Marshall McLuhan, who's a media guru on trivium. And at 5 o'clock, we're going to be talking about two different kinds of thinking according to Carl Jung. So it's a jam-packed schedule. So we look forward to seeing you back very soon. And Sanjay, thank you very much. Great presentation as always. And we'll see you next time. Bye, everybody.
Bye, everyone. Thanks, everyone. Good night.